Hi, good afternoon, everyone. Let's continue with lecture 3. Last class, we were talking about innate immunity. That was that immunity that you were born with. You inherit it from your parents, and it's readily available the moment we're born into this world. And we sort of looked at 5 different mechanisms that are associated with innate immunity, starting with physical and chemical barriers to keep things out of the body. Example of physical barriers include things like skin, mucous membrane, ciliated surfaces, right? Like the airway, for example. We have normal floras as well. These are microorganisms that live on us and in us that helps keep bad pathogens from sticking onto the body. We also talk about chemical barriers, including lysozyme, which is an antibacterial enzyme that's found in saliva, tears, and sweat. There's also low pH in different places in the body, such as the stomach. And then we talk about inflammation. And the key player in the inflammatory response is the mast cell. And these are cells that are anchored to the tissue. They can release the histamine to cause vasodilation, as well as increase the permeability of the blood vessels, both of which is going to help bring more more white cells and more plasma protein to the site of infection. There's also phagocytosis, which is just basically eating up the bacteria, and that's done by neutrophils as well as macrophages. In addition, we also have something called NK cells, the natural killer cells. And these NK cells are there to kill virus-infected cells, as well as tumor cells. And finally, the last mechanism that we talked about is the ability to have a fever. Right? Fever kind of speeds up the cellular activities to help you clear the infection faster. The high temperature also slow down the growth of bacteria and viruses, and it increases the metabolism of our cells so that we can increase the wound healing as well. So that is innate immunity. And like I said, it's something that we're born with. It's not designed to deal with a specific infection, a specific type of pathogens. It's just blanket protection. It's general, non-specific defense. And, it's reasonably well, but it's not adequate. When we do have something that is more difficult to fight off, then we really need to bring out the adaptive immunity. Okay, so let's take a closer look at what adaptive immunity is. So adaptive immunity, also known as acquired immunity, right? Not only does it change over the course of your lifetime, but you also is not born with the adaptive immunity. You have to be exposed to the pathogens in order for you to build the immunity for it. Okay, so if innate immunity mechanisms, as in the phagocytosis, the inflammation, and the fever, do not quickly eliminate an invading pathogen, then we need to have the more long-lasting mechanisms of adaptive immunity. So the characteristics of adaptive immunity. First, it's able to distinguish self versus non-self. It needs to be able to tell what belongs to the body and what does not. Right? Otherwise, it doesn't know what to attack, right? So that's a pretty important function. Just like innate immunity. Right? In innate immunity, they recognize the PAMs, the pathogen-associated molecular patterns on the cells. But for adaptive immunity, it's a little bit different, as we will see. Another characteristic is that it's able to form immune memory once you fought off a particular pathogen, your body remembers it. You're not going to get infected by the same. Well, you can still get infected by the same thing in the future, but you are going to be able to deal with it much quicker the next time. Okay, so for example, like chickenpox, you only get chickenpox once. You're not going to get it again. But as you probably know, people can get the flu multiple times, or they could get COVID multiple times. So like, there is a reason behind that, which we're going to get to. But generally speaking, you do not get infected by the same thing once you have fought them off. Okay, like it may make you feel ill for like maybe a day or so, but you're going to be able to fight it off much quicker and much more effectively in future encounter, because adaptive immunity is able to produce immune memory. Okay, you remember what you fought off. You're going to have these antibodies in your system that will just be ready to deal with it should you be infected by the same thing again in the future. Adaptive immunity, we can also describe it as having very high specificity. Okay, so it's able to create these antibodies that would target a very specific antigens. Remember, antigen is these are just molecules that are found on the surface of a cell, right? And they could be proteins, they could be sugar, and your immune system has the ability to create antibodies that would recognize these antigens. Much like what we learned last week, you're able to create anti-A antibodies, right? So now we're going to be able to create some kind of antibodies that targets these foreign antigens that are on the surface of the pathogens. It's also very diverse, okay? Meaning you have the potential to create antibodies for billions of different types of antigens. Although each antibodies that you create are generally only good for one type of antigens. Basically, you can think of like a set of screwdrivers, okay? Each screwdriver is best used for tightening a particular type of screws. Okay, like if you have screws that have like a cross, then you need to use the right tool for it, right? If you have one of those things with like the hexagons on it, then again you need a different screwdrivers, right? So each screwdriver is only good for a specific type of screws. But if you have the whole set, then you can basically tighten any screws, right? And that's what adaptive immunity is. Individually, each antibody that you make is only good for fighting off a particular kind of foreign antigen, but collectively, right, it's very diverse. You have the potential to make antibodies for all these different kinds of antigens.
So Vincent, is there any specific organs that makes antibodies?
Yeah, that's a good question. So antibodies are created by B cells, right? And in terms of where they make it, we're gonna take a closer look in a bit. Okay, it's not really in a specific organ necessarily, but it can happen in like the lymphatic tissues, right? The spleen, the bone marrows that we talked about. All right.
I have a question, Professor.
Yeah, sure.
So if can you back one slide, please? If, for example, one person had a deficiency of adaptive immunity, what will happen in that person?
Yeah, so we're also going to look at this later on today, but the short answer is they would be very susceptible to getting ill, even from the simplest type of infection. Okay, like you and I probably going to be okay if we catch a cold or something. We'll recover within 3-4 days. But for people without the adaptive immunity, something like a simple cold could make them very, very ill, and without external intervention like medicine and things like that, they're not going to be able to recover on their own. So we call those people immunocompromised, as we will see.
Thank you.
Yeah. So here is antibodies. Like we're not. It's not the first time we talked about antibodies, right? First of all, they are proteins that have a shape that looks like a Y, and they are the key tools for your adaptive immunity to fight off the pathogens. Okay, the ultimate goal of adaptive immunity is to create these antibodies. Now, every antibodies are very similar in the sense that they have a constant region right here. Like the trunk of the Y, that's a constant region right there. Constant region. It's the same for every antibody, kind of like the handle of the screwdriver. Every handle, it's made up the same stuff. It's not unique. The variable region is the arm right here. That's the variable region. And that is going to be the part of the antibody that will recognize the antigens. So for example, we have a orange virus here, and if you look closely, you can see there are little hexagons on the virus, right? So these little hexagons that are on it, that's the viral antigen. Okay? Like we don't have those hexagons on our cells, only the virus do. That's how your immune system know that does not belong to the body. Self versus non-self, right? And so you're gonna make antibodies that have little grooves that would recognize the hexagons. They will bind to the virus, and then we'll talk about what happens once you bind to the virus. But the key thing is you're able to create an antibody that will bind to it. Now on the right side here, we have another pathogen here, bacteria. And on the surface, we have these little semicircles. Those would be the bacterial antigen. Okay, and now you're gonna have to make an antibody that would have grooves that will recognize those semicircles. The antibody I make for the virus will not fit the bacterial antigen. Likewise, the ones that we make for the bacteria is no good for recognizing the virus. That's what I mean by specificity. Generally speaking, an antibody that's created for one type of antigen will not work for another kind. Okay. Now there are a few terms here I want to clarify with you, because maybe I'm not explaining it too well. People often get confused. Okay, pathogens. Can someone tell me what a pathogen is? Just so we're clear, just so we're all on the same page. What is a pathogen?
Bacteria, virus, and parasite.
Yeah, exactly. Like that whole thing, right? Okay, bacteria, virus. Now, antigen. Okay, what are antigen? Antigens are just molecules on the surface of a cell. Our cells have antigens. We learned about it last week. Like type A people have A antigen on the blood cell. Rh positive people have Rh antigen on the surface. So antigen in and of itself is not bad or anything. They are just things that are found on the surface of a cell. The antibodies that we make will target foreign antigens. Okay, so these are, again, like molecules on a cell. It's on your cell, it's on my cell, it's on a bacterial cell, it's on a virus cell. Okay, well, virus is not really a cell, but then like on the surface of a cell, on the surface of the viral particle, you would have these antigens. And your adaptive immunity is able to make antibodies that would recognize these antigens. Okay, and by recognizing the antigens that does not belong to the body, the antibody will be able to tag the bacteria or tag the virus. Is that point clear? Please ask questions before we move on, if it's confusing. Okay, so what does the antibody do? Well, once you create the antibody for a particular virus or bacteria, it's going to take some time to make it, right? It's going to take like a week or sometimes longer to make the antibodies. But once you have the antibodies, the antibody will be able to neutralize the virus or the toxin. Now, toxin are these poisons that are made by bacteria. Sometimes it's not the bacteria that makes us ill, but the poisons that are created by the bacteria. You might have heard of botulinum, right? It's the same toxin that allows people to create the Botox, the anti-wrinkle injection, but it's actually a toxin that causes muscle paralysis. Okay, so it's not the bacteria themselves that directly causes the paralysis, but the toxin that they produce. And what the antibody does is it will recognize these toxins and will neutralize them. So for example, we have these little triangles here that represent the toxins. If you expose a cell to the toxins, then it causes the cell to be damaged. That's the normal scenario. But if you have the antibodies will bind to the toxin and it will make it it will neutralize them basically, and the toxin will no longer damage the cell. That's basically what it is. Over here we have some red cells. If we have antibodies, then the antibodies will bind to the viruses right here, and the virus will not be able to enter the cells. So basically, that's what neutralization means. It neutralizes the threat. It makes the virus, it makes the toxin harmless, basically. It's also possible for antibodies to coat the target, right? Much like complement proteins that we talked about last class, they coat the target, making it easier for phagocytes to identify. Okay, and then they're going to be able to come along and eat them up much faster. Okay, so that's like what an immune complex is. The immune complex is basically antibody, right? Antibodies stuck to their target. Okay, it's the clumping that we learn about, right? It makes it much bigger. It's much easier to identify them and then collect them, eat them up. All right.
Excuse me. So antibodies are fighting antigens or pathogens.
Yeah, so that's where the confusion is. The pathogen is really big, okay? But
Okay.
on the pathogen, there are foreign antigens, right? There are molecules on the pathogens that's not on our cells. That's why we call them foreign antigens. So the antibody will recognize those antibodies. The antibodies will recognize those foreign antigens and it will bind to it. When the antibody binds to the foreign antigen, essentially they are binding to the pathogen as well.
Oh, okay.
Because
Okay.
the
So,
pathogen contains the foreign antigens, right?
okay. So let's say someone with a cold, like sneezes on my face, like my children. Does that mean the pathogen, like that? Am I getting the pathogen from them, like that?
Yeah, basically, right?
Okay.
When they sneeze on you, then you might breathe in whatever virus they have, and it goes into your body, into your lungs,
Yeah.
for example. And generally, the innate immunity may be able to clear them out, and you're fine.
Okay.
But if it's something worse, like a cold, like a flu, like COVID, then your adaptive immunity needs to kick in, right? To produce
Yeah.
antibodies that would specifically deal with the flu virus, for
The flu
example.
virus. Okay, so the flu virus is an antigen.
The flu virus is a pathogen, but on the flu virus, there are different viral molecules, the little spikes, the little triangles, and whatever shape that we don't have. So the body is going to make antibodies that would target those things.
Okay. So pathogen is like a group, and an antigen is like subgroups, like that.
And let me think about this. Uh, hmm. It's not so much a group and a subgroup. It's hold on, let me try to think of an analogy for you, okay? Give me a second, okay? Okay, this is a loose analogy. I don't know if it's gonna help, okay? Like, think of fruits, okay? Apples, bananas, orange, whatever, okay?
Okay.
If I don't show you the whole fruit, I just show you a bit of their skin, are you gonna be able to recognize them?
Yeah, I should.
Yeah, I also, right? Like if I give you a little bit of like a yellow piece, right? Then you probably get, oh, that's the banana, right? You know what I mean?
Yeah, right.
So the antigen is like a piece of the whole fruit. The antigen is like
Oh,
a piece of the
okay.
whole bacteria, right? So if you
Okay.
recognize a little piece, you recognize the whole thing, right? Yeah, I
Okay.
don't know if that helps.
Yeah,
That's the
yeah,
best I
thank
could come
you.
up with on short
Thank
notice,
you.
okay? Yeah. Okay. Another thing the antibodies can do is the antibodies will work with the complement proteins together. Okay, they can complement each other, they can synergize each other, right? And help them form these membrane attack complexes, which we learned last time as well. Okay, they punch holes in the bacteria, right? So the point I'm trying to make is that the adaptive immunity and the innate immunity, they don't work in isolation. Many times they kind of feedback on each other and they kind of strengthen each other. They help enhance each other. Okay, so that's what antibodies are for. Now, the next thing is gonna be a little even more confusing. Okay, on top of the antigen and the pathogen and stuff like that, there's another thing called epitope. Okay, it turns out the antigen is still quite big for the antibody to recognize as a whole. So many times the antibodies will only recognize a small portion of the whole antigen. Okay, so these specific regions on the antigens, they are called epitope. Epitope. Now the reason I bring this up is because the antigen is big, the pathogen is even bigger. When we make the antibodies, we need it to tag to a part that is very unique to the pathogens. Okay, otherwise the antibody is not going to be very effective. Now let me try to play a game with you to help you understand this. All right, and I mean you don't have to unmute and tell me. I'm sure you get the answer. Just think about the answers in your head, okay? I'm gonna describe an animal and you're gonna think about what animal I'm describing, okay? So this thing has a long neck, okay? So what animal am I thinking of? Hopefully you're thinking of giraffe, right?
Giraffe.
Yeah, very good, okay? Now this next animal has a very long nose. Okay, people call it the trunk. And hopefully you're thinking about the elephant, right? Okay, so far so good, right? How about the next one? How about this animal has a long tail? Has a long tail. Now what am I thinking about? And you're like, I'm not sure. It could be many things. Many things have a long tail, right? Like, I don't know, like a lion has a long tail, a mouse has a long tail, even snake has a long tail, right? And it's not a defining feature of an animal. So it's hard for you to guess, right? The long neck, the long nose, those are defining features, right? And that's what I mean when I say you need to create an antibody that is very unique to a particular virus or bacteria. Those tend to be the most effective kind of antibodies. So for example, again, I'm just making this up. Let's say there's A, B, and C on this pathogen, and then there's like C, D, and F here, and then F and A and B here, right? So let's say I'm infected with this guy. I'm gonna make some antibodies. So I can make an antibody that targets C. That's like an anti-C antibody. I can make one that's targeting D or make one that's targeting F. So 3 types of antibodies your body is going to be able to make, but which one is the most effective? Is it the C, the D, or the F? What do you guys think?
D.
Yeah, because D is the only one that's found in this particular one, right? So that tends to be the best antibodies to make. So long story short, when people create a vaccine, they have to select, and we're going to talk more about vaccines later, but especially with the mRNA vaccines, you're making a portion of a protein, right? So you want to be making a portion of the protein that is going to create the most effective antibodies, okay? Something that is very unique for this particular virus or bacteria, okay? It needs to be very immunogenic, okay? So basically the epitope is the part on the organism, the part on the antigen that is going to uniquely identify the organism. All right, let's take a look at this one. Let's see if you guys feel like doing more polls today. It's hit and miss. Some days, lots of answers. Other days, not so much. Okay, no answers at all. That's okay. We'll just take this up. I'm just looking at the answers. Some of them we actually haven't really talked about in details. Maybe that's why you guys are not really sure what the answers are. But let's see. All the correct one. Antibodies are Y-shaped protein. That's true. Right? At least you know that one. You should know that one, right? We talk about that. Antibodies produced against one pathogen will generally work against another pathogen. That is not true, right? What you make for the red bacteria is not going to work for the orange viruses, right? Usually, it won't. Okay, keep in mind, usually, right? We're going to come back to this point later. Antibodies are produced by plasma cells. That's true. I'll tell you about plasma cells in a bit. Antibodies are found in blood plasma. That's also true. We learned that last week. Variation in antibodies arise from gene shuffling. That is also true. And I will tell you more about gene shuffling in a bit. Okay. So, you know, some of the answers we haven't talked about, like C and E, but at least you should know A and D, right? But hopefully after the note, it will make more sense to you. Okay. That's all. I hope you're ready for a super long note. I'm just going to continue from last week's last class notes. I haven't posted that one yet because it was just so short, and I wanted to add to it. Okay, so now we're gonna write another note called antibody. You need color for this one, okay? Definitely color. Without color, it's not gonna make any sense. Antibody production. Okay, just to give you an idea what's gonna be happening in terms of space, I'm gonna start here, and then I'm gonna go over here, and then I'm gonna go here, and then they will all be connected in the end, okay? So like, just working my way from the left to the right. All right, here we go. Just stop me anytime, okay? You are not interrupting. It's okay. Just speak up anytime you get lost. Anytime you get lost. Okay, so we talk about innate immunity, right? Innate immunity. Last time. Just, you know, for the sake of revision, let's list them again. We have things like physical and chemical barriers to keep things out. And then we have inflammation. If they do come in, it will trigger inflammation. Inflammation will hopefully help clear the infection. We also have phagocytes. And we have NK cells and fever. So I want to focus on the phagocytes here. The phagocytes are particularly important because they serve.
Vincent, sorry, before you go on. So when you say fever, so does that mean fever is like not a bad thing?
Yeah, usually it's not a bad thing. Usually it just means that your body is fighting it off, right? I mean, it's concerning if it's like really high fever, like over 40, especially in kids, then you really need to take them to the emergency and see what happens. But generally, low-grade fever, especially if there's no other symptoms, like if the person feels fine otherwise, if the kids is like, still running around, then usually it might not be a cause for concern. So phagocytes are particularly important because it is a link between innate and adaptive immunity. It's a link between innate and adaptive immunity. So what I'm going to do is I am going to have some bacteria here. Now, I just keep on using bacteria as an example because most people can relate to that better, I guess. But it's the true for, with virus and stuff like that. Okay, so I'm just using bacteria as an example. Okay, so here we have some red bacteria. Okay, I don't know where we caught it, but we caught some. Okay, and it has entered our body. We got some inflammation going on, and then the phagocytes are working as best as they can. And maybe we have a bit of fever, maybe not. It doesn't, it's not a fever is not a requirement to trigger adaptive immunity. Okay, so let's take a phagocyte here. Okay, and I'm gonna use macrophage. I'll explain why in a bit. Okay, macrophage. Again, you have to use color with me. Okay, otherwise your note is not gonna make sense. So what's the macrophage gonna do? Well, I'm sure you know. The macrophage is going to eat the bacteria, right? Because the bacteria has certain signals on them that it's begging the phagocytes to eat them. So then it's gonna eat them up, and it's gonna put it in a vesicle, for example. There. Okay. There might be some other ones here floating around, waiting to be eaten. Okay. So this process is called phagocytosis. Nothing is new so far. Everything is, it's something we talked about before. Okay. Next is new. Next, what's going to happen is the macrophage is going to do something called antigen processing. Okay, don't look this stuff up on the internet. Okay, I am simplifying it a lot. If you read it on the internet, it's going to confuse you. Okay, just take my word for it. I will only test you what I'm teaching you. Okay, and that should be enough. So what is antigen processing? Antigen processing is the macrophage. Macrophage means big eater, huh? The macrophage will break down the bacterial antigen, okay? And it could be virus, it could be bacteria, it could be other stuff, but it will break down these foreign antigens and display them on the cell surface. Right, so here is our macrophage again, and then it's putting bits and pieces of the bacterial antigen on its surface. Okay, it's like, hey, look, I just ate this red bacteria and it's infecting our body. Hey, look, 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 that's what they look like. Okay, why don't you go find them and eat them too? Make sense? That's called antigen processing. This is for real. It does happen. Okay. There we go. Any questions so far?
So this message is for who?
It's for who? Yeah, okay. I'll show you in a bit. Yeah, very good question. Yeah, I'll show you. Like who are we showing it to, right? Okay. So let's see.
Yes.
All right, so we're gonna move over here for a bit, and I'm gonna draw something else. Okay. Despite my bad drawing here, do you know what structure I'm trying to draw here? Like what is that? What should I label that as?
Bone marrow.
Yes, very good. This is a bone, and then the inside stuff is the bone marrow. And what's going on in the bone marrow? Well, bone marrow is where you produce the blood cells, right? Okay, so coming out from the bone marrow, you know, maybe I should put this up here. Coming out from the bone marrow is going to be a type of cells that we call naive T cells. Okay, naive is spelled with like a special I. Okay, like with 2 dots. Sometimes you see that in some words. Okay, so a naive T cells. Let me give you a little bit of description for it. Each cell, the naive cells, has a unique receptor. Okay. The receptor is created by gene shuffling. Okay, and I'll explain what gene shuffling is. Do you remember from last semester what exon and introns are? Do you guys remember that? Like, which one do we keep? The exons or the introns? Does that mean anything to you guys? The introns, exons? No? Okay, so I'm not going to explain that. It's not important. The introns are the things that we cut out. The exons are the ones that we keep, right, in the process of transcription. Okay, but gene shuffling works something like this, right? You have a bunch of genes in your DNA that is dedicated for immunity. Okay, it's like having a deck of playing cards. You have 52 cards, and every time you just deal out 6 cards. Okay, so when you do it for the first time, that 6th card is probably going to be different from when you do it the second time, right? And when you do it for the 3rd time, it's going to be different, right? So that's what gene shuffling is. You have a set of genes, but depending on which genes you take each time, the result is going to be different, and that is what allows you to create different receptors. Because the receptors are proteins, right? Okay, they're membrane proteins, which are coded by the genes. So, you take different set of genes, you end up getting different shaped receptors.
Vincent, this gene is not to be one for reproduction, right?
Say that one more time.
This gene, like, is not the reproductive gene ones.
No, but they are a set of genes that are specifically designated for immunity for this particular purpose.
Okay.
Yeah. Okay. So, in reality, it's not just like one deck of cards. It's more like, I don't know, like 10 deck of cards all mixed together. And then you are dealing out 10, 20 cards, each time. So virtually you have billions and billions of different combinations, right? And that's what I mean when I say adaptive immunity has the potential to recognize billions of different types of antigens, okay? But each time, we're only going to be able to deal out like a specific hand. So visually, what does this look like to help you understand? Well, we have a naive T cells here. We have naive T cells. Okay, so let's say we just draw 4. Okay? But again, in reality, you have a pool of these naive T cells. Each one of them is going to be different. So, you know, these naive T cells might have a blue receptor. These naive T cells might have a green receptor, and the next one might have a red receptor, and then the next one might have like a purple receptor. Okay, I'm using color to help you understand the difference, okay? But again, like in reality, they are just membrane proteins that have different shape, okay? Like which shape is gonna match the bacterial antigen. That's basically what we're looking for. But it's much easier to use color than to use shape. Okay, so these are naive T cells, and again, this is a receptor for recognizing antigen. Okay, and I don't know if you remember, but like at the end of lecture 2, you know, when we talk about the thymus, I said the T cells, they have to do a auto-reactive test, right? So like the receptor, if it recognizes our own antigens, we got to kill the naive T cells. They're not allowed to come out. Okay. Now, another quick word, naive. Naive means they have not been activated yet. When we say someone is naive, it's like, you know, you haven't, like kids are so naive, they don't know the dangers of the world, right? It's kind of like that. The naive T cells, they haven't been exposed to the pathogens yet. So they are naive. They're not ready to fight yet. Okay, so Fatima, you were asking earlier, like who are we what like the macrophage, like who are they presenting these antigens to? Okay, and then I think you have the answer now. This guy's gonna come over here, and it's gonna do what's called antigen presentation. That's again what it actually is called. And so antigen presentation involves the macrophage will show the bacterial antigen, the little red blobs on the surface, to naive T cells. Okay. So, you know, stop copying for a second and just try to listen to what I have to say first. So the macrophage will come, and then it will show it to this naive T cell with the blue receptor. It's like, hey, we're being attacked by these bacteria. They look like these red little knobs. Do you have the right receptor to fight them? And the answer is nope. Ask the next guy. So they're going to present to the next one. You know, do you have the right receptor? Nope, I got the green receptor and we need the red one. So that's not going to work. And then it's going to show to this one. And this one is the chosen one. Okay, that's the selected one. So let me pause there, let you catch up, and then I'll tell you the rest of the story.
Can you repeat, Professor?
Sure. So you understand, do you understand what these little red knobs are on the surface of the
Yes.
cell? Yeah.
Yes.
Yeah. So that we need to find the naive T cells that has the right tools to fight these bacteria, right? The first one is not going to be useful. Right?
Yeah.
That one is only good for fighting blue bacteria, so to speak, right?
Mm.
And then so on and so forth. So until we are able to find the right cell.
Mm.
Okay. That's why sometimes it takes longer for you to, you know, get better in some cases, because these naive T cells are randomly generated. It's entirely possible that at the specific moment, the population of T cells that you have none of them has the right receptor. Okay, then you have to wait. You have to wait for the body to create the right kind of T cells.
Yeah.
Okay.
Thank you.
Yeah.
So Vincent, these T cells are released in the blood.
The T cells are in the thymus, and then when they get chosen, then they're gonna go into the blood.
Okay.
Okay, right. So like, if you get infected by these yellow bacteria, let's say, right, then none of these are gonna be able to help. You're gonna have to wait for it. It's like, shuffling the genes, right? Dealing the cards. You're gonna deal the cards until you get the right combination. And sometimes that takes quite a bit of time. And during the time you're waiting, these yellow bacteria is causing you to become sick, right? So it's like a race, right? Like, do they make you very ill before you can find the antibodies, or are you able to stop them, right? So sometimes we don't. The pathogen is so damaging that we won't be able to fight off ourselves. Then we need medicine to help us, antibiotics, for example. Okay. Now, macrophage is the example I use, right? What's another example of a phagocyte? Do you guys remember? I think I typed it in the chat earlier. Besides macrophage, what's another phagocyte that we learned? The most common white cell starts with an N.
Neutrophil.
But neutrophils cannot do antigen presentation. Okay, not every phagocyte can do this. Macrophage can. So macrophage actually has another name. We call it an APC. APC. Okay, I'm gonna put a star here. What's a good color here? I'm gonna put a star here. APC stands for antigen presenting cell. Okay. So they can eat up the bacteria, they can put it on the surface, and then they can present it to the naive T cell. All right, they are chosen. This T cell has been chosen. Okay. Once the T cell, the naive T cells, has been selected by APC, by the macrophage, the naive T cell undergo clonal expansion. That's just a fancy word for mitosis. But again, that's what it's called, okay? Like they are gonna make many, many copies of themselves. Okay, I don't need to make any other ones. They're not useful, they're not helpful in this situation. I only need the ones with the red receptors, right? So we're gonna make a bunch of them. Okay, we're gonna clone all of these guys. Okay, so we end up having like a little army of them, okay? Like you get the point, right? Okay. So there we go. We end up having a population, okay, of these naive T cells. So these naive T cells, now that I have a bunch of them, they're gonna differentiate and become different things. Okay, they're gonna differentiate and become different things. Okay. So one of the things that they can become is called cytotoxic or killer T cell. Okay, so they look like this. Okay, I'm gonna draw the cell. And then, you know, these are gonna go attack things. Okay, so like maybe I give it like a sword or something. Okay. Usually this helps people remember it. Okay. Okay, so these are cytotoxic or killer T cells, and their job is to kill infected cells. Okay. By punching holes. And injecting toxin into them. Okay, you're gonna go kill the infected cells. Okay, that is cytotoxic or killer T cell. They might become, okay, I'm not doing this in any particular order, okay? They might become this guy that's holding a stop sign. Okay, we call them suppressor. Suppressor T cell. Okay. Hopefully, you're going to be able to clear the infection soon. And someone has to stop this clonal expansion business, right? Like you don't want to keep on expanding these white cells. Uncontrolled cellular division is a dangerous thing. We learned that, right? It could lead to cancer. In this case, because it's blood cells, it could lead to leukemia. So we don't want that. So the suppressor T cell will stop immune activity, including the clonal expansion, once the pathogen is cleared. Okay, I'll give you a moment to catch up. Ask me anything.
So then in this scenario, macrophages are working or not?
Yeah, so macrophages is still working in the background, it's still eating as much as possible. But then because the infection has been kind of overwhelming, that's why we need to bring out the adaptive immunity, right? And
Okay.
part of the adaptive immunity involves creating these specialized cells that would help coordinate the attack.
Okay, thank you.
Okay. So this process is called differentiation. It's not the first time I told you about this word. Okay, so the naive T cell will differentiate into cytotoxic T cells, right? They will differentiate into suppressor T cell. Like they gotta choose a path, okay? Like once you become, there's no coming back. Once you become a cytotoxic cell, you're gonna stay as that, okay? We have another one here. And you have to know these cells, okay? This one is, I'm gonna put like a little bubble here. Okay, I'm gonna put the bacteria in there. This guy is called the memory T cell. Okay, this is the cell that will stay in your body for pretty much ever, in case you get infected by the same thing in the future. So next time you get infected by the red bacteria, no worries. I remember fighting it off before. We are gonna deal with it much quicker and with a much better efficiency. So stay in the body for a very long time, right? In case of future infection by the same pathogen, same bacteria, same virus, whatever you have. Okay, so for everything that you have fought off previously in your lifetime, you're gonna have a memory cell somewhere in your body. Okay, you have fought off COVID, you got some memory cells that remembers COVID. Now I know some of you are thinking, then how is it possible for you to get COVID again? We're going to get to that, okay? We're going to get to that. All right. The last kind of cells that that the naive T cells can become, okay, and I want to draw it up here because I'm gonna connect it with the bone marrow in a bit, okay? These are called helper T cells. Helper T cells. Okay. I'm just gonna give them a really big hand. Okay, because they love to help things with these big hands. If you think of a better symbol, let me know. This is a helper T cell. Okay, and what does helper T cell do? Well, coming out of the bone marrow, the naive T cell is not the only thing. We also have naive B cells. Okay, and similar to naive T cells, the naive B cells have unique, in this case, antibodies. Okay, so you might have a naive B cells that has a receptor for like that's capable of making like green antibodies. You might have one that's capable of making blue antibodies, and surely enough, we have one that's making red antibodies. Okay, and then you might even have one that makes yellow antibodies. So here, let me write down for you. Each has the ability to make a specific antibody. And what the helper T cell will do is it will go and select the right tool. Like this guy can't help, this guy can't help, this one get selected. So it will choose the naive B cell that has the correct antibody. Okay.
So Vincent, that means the naive T cells and B cells, they work hand in hand.
That's right. Yeah, but there is a kind of like an order to it. Okay, to get the B cells, we need the T cells first.
Okay.
Yeah. So once it is selected, again, we have clonal expansion. Okay, which means you're gonna get a bunch of them. Okay, you're gonna get a bunch of them. Okay, I think you get the point. I'm not gonna go crazy like before here. There we go. And they're gonna differentiate again. You're gonna differentiate. But the B cells don't have as many choices. Right? They can only become two things. Right? They can become memory B cell, which is the same job as the other guy, as the memory T cells. Okay, they also remember. Okay, they also remember. Okay. But they have all these antibodies on them. Like they're like, okay, next time I see you, I'm gonna like mass produce these antibodies. I'm not gonna write a description. It's the same description. Okay. And then we have plasma cells, which is what you saw in the poll question. And the plasma cells are basically antibody factories. Okay, so they will make a ton of antibodies that will help kill the bacteria. Okay, so these are going to mass produce antibodies. Now, how are the antibodies going to help? We learned about it in the PowerPoint slides, right? Just going to verbally recite it for you. They can neutralize viruses. They can neutralize toxins. They can form immune complex, which will help help with phagocytosis. They will activate complement proteins, right? So everything that we talk about in the slides, that's what the antibodies will do. And hopefully all these actions collectively will help us clear the infection. Okay.
Listen, can you bring up the
Yeah.
going up?
Higher?
So here, if the T cells don't find the right like antibody,
Oh, like here?
what? Yes.
Yeah. So again, like it's by chance, right? More likely than not, you will have something. Okay. But if you don't, then you're because these populations keep on overturning. Okay, like the ones that are not used, they get killed off, and then you get new ones. You constantly refresh the population. Okay, so like it's just a matter of time before you are able to create the right cell, right? But that's the key thing. Like, are you able to make it in time before it makes you super ill, right? Does that make sense?
Yes, thank you.
Yeah. Okay. So I know I just used the bacteria as an example here, but it doesn't have to be bacteria. It doesn't even have to be viruses. It could be, this could be a Rh negative person, and then instead of bacteria, we have Rh positive cells. Okay. And then your body's gonna go through all these things, and ultimately, what you end up getting is anti-Rh antibodies. Right. And then you have these memory cells in the body. That's why, like, you are sensitized now. In the future, you cannot accept positive blood again because you already have the memory cells in you. You already have the antibodies in you. You see how the things are like all connected?
Vincent, so the T cells have the colored thing as the receptor for recognizing antigens, right?
The T cells, did you say?
Yeah, the T cells.
Yeah.
Okay, so when they recognize these antigens, they undergo cell division and like mitosis, right?
Okay, yeah, but they have to be selected. They have to be chosen first.
Like yeah.
Right,
That's
right.
chosen, right? Yeah. So once they are chosen, once they have been divided and then they've been multiplying and having then at what point do they now say, okay, we are bringing the B cells?
Oh yeah, it's like so it's hard to it's really hard to put them in chronological order because a lot of these things actually happen like simultaneously. So at the same time that it is becoming the cytotoxic T cells, some of it is already becoming the helper T cell, and then almost immediately they're gonna have to choose the naive B cell.
Okay.
Yeah, because time is of the essence, right? Like you gotta make those antibodies as quickly as possible. Otherwise, the longer we wait, the more damage the pathogen is causing the body.
So only B cells can make the antibodies.
That's right. Only the B cells can become the factories, the plasma cells. That's right. So like on the quiz, on the test, if I ask you what cell produce antibodies, you have to select plasma cells. Plasma cells. Okay, just a couple more things here. Here, let me summarize it for you, okay, in another way. I know it's the same information, but some people see it a little bit better if I draw it out like this in a linear fashion. So we have the naive T cells. Again, you can just use one dot. I'm just showing you because that's how textbooks do it. Okay, naive T cells. What do we need to activate the naive T cells? What should I put up here? What kind of cell do we need to activate them? Look at the picture.
Macrophages.
That's right, the macrophage, right? Okay, but it doesn't have to be a macrophage. It could be any APC, right? APC, antigen presenting cell. But you've only learned one from me, which is macrophage. Right? And then once they are chosen, once they are activated by the macrophage, then they could become 4 different things, yeah? 4 different things. Okay. So this process is like differentiation, right? And I'm just gonna list them again. Cytotoxic T cell, cytotoxic or killer T cell, same thing, okay? Don't confuse killer T cells with the other killer, right? What's the other killer that I'm talking about?
NK cell.
NK cells, natural killer. They are part of innate immunity. This kind of killer is like from the T cell, right? That's why sometimes people just say cytotoxic. Okay, they kill infected cells. And then we have the suppressor T cell. And then they have memory T cells. And what's the other one? Helper T cell, right? Okay, so these guys, they are the effectors of T cells. If I ask you what are the effectors of T cells, I'm asking you what will the T cell become? Okay, they will become all these things. And this branch of adaptive immunity is called cell-mediated immunity. Okay. Cell-mediated immunity. That's the T cell branch. Now let's not forget our other branch, which is the naive B cell. And the naive B cell needs the helper T cell to select them, right? To activate them. Once activated, they can, like, what are the effectors of B cells? The effector of B cells would be plasma cell and memory B cell. These are the effectors of B cell. And this branch of immunity is called antibody mediated immunity. Okay, sometimes it's also called humoral immunity. Okay, that's the whole story. Right? Again, I already condense it as much as I can, make it as simple as I can, but this is the minimum amount that you have to know. That's how your immunity works, okay? That's how you make antibodies. Questions before the break? I know. It's a lot of information, which is why I told you, like, this lesson is the hardest, right? This lecture. Yeah, but you know, hopefully, it's just one step leading to the next, right? And ultimately, we need to make the antibodies. Okay. All right. I'll let you guys dwell on this a little bit. I cannot fit everything onto the page. I guess I'll just make it smaller like that for you to see. But we're gonna come back, okay? In 5 minutes. Then we're gonna take more notes. So see you in 5. Okay. We're gonna keep going. Any last minute question before we move on to another topic? We're almost there. Okay. Next, we're gonna talk a little bit about vaccines, but I just want to do some quick polls with you guys to see what you think about vaccines, because there are a lot of misconception here. Just interested to see what people think. Okay. That is what a lot of people believe here. Do you think it's true or false? It is possible to get the flu from the flu shot. What do you think? Again, like, just tell me what you think, gut feeling. Would be good if we can get more responses, so it's more representative of you know, the whole class. Anything else? Last call. Okay, so here are the results. You know, we only have 7 people participated, but then 4 of you think it's true and 3 of you think it's not true. This is not true. Okay, this is false, 100% false. It's not possible. It's not possible to get not just the flu from the flu shot. You're not supposed to be able to catch the disease you are getting vaccinated for. If you're getting vaccinated for COVID, you're not going to catch COVID from it. Okay? If you're getting vaccinated from for chickenpox, you're not going to catch chickenpox from it. Okay, so the flu shot is something that has what we call an attenuated virus. An attenuated virus is a virus that can no longer cause sickness. Right? It's basically just a shell without the parts that can make you ill. Right? So, you know, it's because it's just a shell, it's not possible to cause the actual disease. Now, sometimes you hear people say, but I got the flu shot like 2 days ago and then I have the flu now. Right? It must be from the flu shot. In those instances, you got to remember the flu shot does not work immediately, right? The flu shot is just a way to educate your body and help trigger the process of creating antibodies for the flu. You know, it's like giving you the shell. It's like, hey, that's what the flu virus looks like. You better make memory cells and antibodies for it. You are not going to be able to do it immediately. This whole process takes a week or so, right? So you're not going to immediately get protection. Maybe you catch the flu during that period, right? But not from the flu shot, maybe from someone else. Or maybe you already caught the virus before you got the flu shot, right? And then it's just a coincidence. So not possible to get the flu from the flu shot. But don't take my word for it. Do your own research and find out. But I can tell you it's not true.
Well, Vincent, I thought, you know, like when we get sick, like the popular chickenpox example.
Sure.
Yeah, so I like if someone like me that doesn't have chickenpox before, you know, like just giving me the vaccine, how does my body, you know, like know that okay, this is for the chickenpox?
Oh, because
I thought my body was going to be, you know, what's the word again? Sensitized. Yeah.
Sure. So like the vaccine contains parts of the virus, right? So that's how you know. Like if they're gonna make the chickenpox vaccine, they're gonna use part of the chickenpox virus. Okay. To again educate your immune system. So when the real thing comes, you will be ready. You already have the memory cells and the antibodies for it, right?
Okay, so the one that would be given to me would not it would not cause me to have the sickness at all. Okay.
Exactly. That's right. Okay. It's not possible because again, the virus that they use is either attenuated, which means weakened, or they are dead altogether. Okay. So not possible. How about this one? Okay. This is one that I hear a lot during the pandemic. Let's see. What do you guys think? Again, right? No judgment. You just choose what you think is right, and then I'll tell you the answer. Anyone else? Oh, more participation this time. Good. All right. Yeah. This is not true. This is not true. Okay. So if you remember from last semester, I hope you remember at least something from last semester when I taught you all these things. We know DNA becomes mRNA and mRNA becomes protein, right? The mRNA vaccine basically uses the code, the mRNA, and then your APC, we know what APCs are now. The mRNA is going to go into the APC, mRNA, and basically the mRNA is going to get translated into proteins. In this case, the viral proteins from the COVID. Okay, and then it does the presentation and so on and so forth. Then you create the anti-COVID antibodies. That's what mRNA vaccines do. It doesn't change your DNA in any way. You just feed the mRNA is actually surrounded by a lipid coating. Okay, as you learn, the cell membrane allows hydrophobic stuff to go through, right? So that's why the lipid surrounds the mRNA is going to deliver it to the APC. Then the APC will have all these viruses particle on the surface. Okay, it's the same process as this, exact same process. The only difference is rather than doing the phagocytosis and then antigen processing, we are feeding the APC the exact sequence that we want them to make, which is why mRNA vaccines are very powerful. And with the right code, with the right mRNA, we can put theoretically, we can put any antigens on the surface of the APC and allow them to trigger an immune response to it. Right? So let's say there's a new, it's not new, okay? There's a flu virus that's circulating right now. It's infecting a lot of cows in the States. Do you guys know what I'm talking about? It's on the news recently. It's like 4 letters, 2 letters, 2 numbers. You guys know what I'm talking about? No? There's something called H5N1. Does that ring a bell to anybody? It's a flu virus. Yeah? Yeah, so it's a flu virus. It's an avian flu. It is avian because it primarily affects birds, okay? But occasionally they can go into mammals, right? Okay, so what experts are really worried about is that this H5N1 might go into a mammal, like a pig, for example, and inside the pig, maybe it also get infected by like a human version of the flu virus, and then the 2 virus can exchange genetic material. Then all of a sudden you might have this new influenza virus that's very deadly and it's able to spread from people to people, right? Then we might have another pandemic. But with the mRNA vaccine, right, maybe we can put these H5N1 viruses on the surface of the APC, and then we're going to be able to, vaccinate very quickly. All right, moving on. Vaccine contains live viruses or bacteria. I told you that's possible, but not every vaccines have live viruses, okay? And even if they're alive, they are attenuated, they are weakened, right? They cannot cause disease, right? So this is like sometimes true, okay, but not for all. Right? Many of these vaccines don't have live viruses in it. Sometimes they only contain parts of the virus. Right? So it depends on which vaccines we're talking about. Typically, live vaccines are the most effective because they are the closest thing to the real thing. Okay? But live vaccines are not suitable for everybody. People who are pregnant, for example, people who are undergoing chemotherapy, they're not suitable to receive live vaccines. Okay. This one is like, very, very common you hear. What do you guys think? What do you guys think? Any last call? I have my usual 7 response. Any more? Okay. Long story short, this is false. This is false. Okay. The reason why this was a thing is because I forgot when, but like maybe 10, 15 years ago, or maybe even longer, 20 years ago, there was a scientist, medical doctor / scientist, who published a paper linking the MMR vaccines, the mumps, rubellas, and measles vaccines with autism. Okay, so that paper went viral, and many people thought that there's a link between the vaccines and the autism, and that doctor actually went around the States and then give talks about it. But then later it was found out that he faked the data. Okay, he totally fabricated the data, the results. It's not true. So they retracted his studies, and since then there are like numerous studies that have tried to look into the connection between the vaccines and the autism, and they found no connection at all. Okay, but the damage has already been done. Okay, it's just the way that humans are, right? Like we are more in tune to like negative news than to positive ones. Okay, like sometimes we feel like, oh, we might as well believe the worst outcomes, because what if it's true, right? And then we ignore the fact that there are actually literally hundreds of other papers that have disproved this. So short answer, it is not true. But like I said, don't take my word for it. Do your own research. Okay, and be careful where you're looking it up. There are websites out there that are dedicated to anti-vaccines movements. Those might not be the best place to gather your information. All right, back to our regular schedule program. Another note, type of adaptive immunity. So here we go. This one is not too bad. Okay, this one is just gonna take, I don't know, maybe 20 minutes or something. So here we go. Type of adaptive immunity. Okay. Middle of the page, please. And then we're gonna separate it into 2 categories. So over here we have what we call the which one is it? I'm just thinking which one to do first. Let's do passive immunity. And the opposite of passive immunity is, of course, active immunity. Okay, so passive immunity means the host is us, the person that's getting infected, okay? The host does not make antibodies. Rather, antibodies are given to them. Okay, so you don't make your own antibodies in this case. You just receive them. Someone else made it, or something else made it. You are just gonna receive it. You're just gonna take it. Because you did not make the antibodies, what kind of cells are you not making?
B cells.
Specifically, what kind of cells?
Plasma cells.
Yeah, and I guess the thing I'm looking for is memory cells. Okay, so no memory cells are made. Therefore, it is short-term, but immediate protection. Okay, short-term, but immediate. So just like, you know, studying for the test, if you don't study and you just cheat, someone just give you the answer, you're not going to remember the answers. Okay, it's just going to be short-term, but you will get the answer right away. So that's passive immunity. For active immunity, the host makes their own antibody. Do you make the antibodies right away? Do you make antibodies right away?
No.
No, it takes like some time, right? So therefore, this is you make memory cells, so it's long-term protection. Okay, but not immediate. Okay. All right. So let's give some examples. Under passive immunity, there are two kinds. We have natural and we have artificial. Another word for artificial is induced. So we use these terms in combination with each other. Okay, we would say natural passive immunity, or we say artificial passive. So natural is, you know, what exactly what you think it means. Okay, it just happens naturally. There is no, you know, medical intervention. Okay? Something that would just happen, you know, like it could have happened like 500 years ago, naturally. Okay, there's no medical intervention required. There's no injection of any kind. So who can we get antibodies from? Naturally. Anybody have any guess? It has to do with something we talked about already before, last week maybe.
The parents.
That's right. You can get it from mom, right? Okay. So mother can pass antibodies to their babies, to her baby, through the placenta. And how does the mother give the baby more antibody once the baby is born? What do we need to do? Baby.
Like by breastfeeding?
Exactly, and breastfeeding. That's why some people say breastfeeding is good for the baby, because you can transfer antibodies to the babies, right? We already know antibodies can go to the baby through the placenta. That's what happens with the hemolytic disease of a newborn, right? Except in this case, we're not talking about harmful antibodies. We're talking about protective antibodies. During the pandemic, my sister was pregnant, and she was not sure if she should get the COVID vaccine. Okay, worrying that it's going to affect the baby. But then her doctor said, yeah, you should get the vaccine, because if you get immunity for the COVID, you actually can pass those immunity to your baby. Right? That's example of natural passive immunity. You understand? There's nothing artificial about this. Mother to baby, natural. Now, remember, this is short-term, but immediate. So once the antibody goes into the baby will have immunity. Right? Let's just say, use the example, right? Like, you know, the mother is immune to COVID. Some antibodies go to the baby. So the baby is born with the COVID antibodies. Okay. And then, you know, through breastfeeding, maybe there's some more immunity being transferred, but there's no control as to what actually get passed through, right? Okay, like it's not like you can pick and choose, right? It's, again, like the exact mechanism is not well understood, but you're not sure what actually goes through. The point is, if the mother stopped breastfeeding, then the baby loses the immunity, right? The antibodies that the baby receives is not going to last forever. Right? Eventually, the baby will grow up, they will have to get their own immunization, or they will have to make their own antibody. Is that okay? All right, next one. Artificial passive immunity. Okay, so there are a couple of examples here. Okay, we can use it as a protection. Protection. Protection / prevention. Prevention. Okay, so this would be example would be like using a serum. Serum is like purify antibodies. From an immune individual. Okay, so maybe, let me give you an example. Let's say I have to go on a trip for work, okay? Like I got to go in 3 days. Where I'm going, there is this disease, okay? I don't have protection against that disease. It's not enough time for me to get traditional vaccination, because even if I get the vaccination, I just told you earlier, you're not going to get protection right away, right? Like it's going to take a week or so before my body makes the antibodies, right? But I got to go in 3 days. Right? So what do? Well, I can get a serum. Right? So someone else who had immunity for that disease X, right? We can purify the antibodies from those people, and then I receive the antibodies. The antibodies are injected into me directly, and immediately I will have protection. Okay, but it's not going to be long lasting. If I come back for my trip, and then, you know, in half a year, if I have to go again, I might need another serum, unless I, you know, get vaccinated in between, right? So that's what a serum is for. Understand? Okay, it can also be used as treatment, or what's the word I'm looking for? Like prophylaxis. Okay, prophylaxis means like, you know, just in case. Okay, I'll give you an example. E.g., this is real life example. Okay, so like this, what is this? Almost 2 years ago, I think. Two years, yeah. 2 years ago, I was just running. I run regularly, okay, a couple times a week. So I was just running on the street, and there was a person holding 2 dogs walking towards me, and then, you know, like I didn't think of anything of it, or I just keep running. But then as I ran past the dogs launched at me and then bit me right in the gut, okay, in the abdomen. Okay, so you know, it hurt a little bit, and then I look at the person, and then I didn't want to disrupt my my running rhythm, so I just kept going. Okay, and then when I got home and took a shower, it was bleeding, like the wound was bleeding. So I was like, shoot, right, this is not good. What disease am I worrying about? Does anybody know?
Rabies?
Yes, rabies. So I called my doctor. I was like, hey, I got bitten by a dog. And then he's like, did you take down the information of the owner? And I was like, nope. And then he's like, oh, you should have. because, you know, even though the dogs are supposed to be vaccinated and all, but you don't know. So you need to, you know, it's unusual for a dog to bite a person. So, you know, if the dog actually has the rabies and it exhibits some symptoms, then you know, at least the owner can contact you and then we would know, right? But now we don't know. There's no way to find out. So you got to come in, because if the rabies symptoms starts to show, then there is like a 99% chance that you're gonna die. And then I was like, okay, you got me convinced. I'm coming in, right? So he actually had to contact public health and order the serum for me. Okay, so the serum is anti-rabies antibodies, basically. And so he injected me with the anti-rabies antibodies. It was like 2 shots directly into the wound. He also gave me a shot for tetanus, okay, because apparently the dog saliva can have the tetanus bacteria. And I had to go back for the whole month, once a week, to get 4 additional shots. So that is an example of a prophylaxis / treatment. You don't know if you actually caught the rabies, but if you did, you know, these antibodies are going to help suppress the virus before it causes a problem, right? So it could be a treatment, right? So like we don't know. In these uncertain situations, you need to have these. So like, you know, anti-rabies treatments. Okay, there is also something called MAB, okay, which stands for monoclonal antibodies. Depending on what disease we're talking about, sometimes they are very effective, sometimes they're not. So like hepatitis or something, like if you guys work in a healthcare setting, and let's say you give an injection to someone with hepatitis and you're supposed to dispose of the needle, right? In a sharp object kind of waste disposal unit. But if you accidentally jab yourself, then that's a problem because now you might potentially caught the hepatitis, right? Then they will have to give you monoclonal antibodies. These are purified antibodies that are specifically designed to fight off the hepatitis virus. Okay, and hopefully, again, it will clear it should you have been infected. And even if you haven't, we're just doing it as a prophylaxis. So does everybody understand artificial induced passive immunity? Any questions?
So
Okay.
this antibody serum, so doesn't stimulate the bone marrow to produce like B cells?
No, the antibodies are already the end products, right? So
Okay.
they're ready to fight, but it will not trigger your immune system. Yeah.
Okay.
Okay, so where do they go? Like, do they go into the bloodstream like immediately?
Yeah, they inject it like directly into your blood. Yeah. Well, at least the one I got, that's what happens. The dog bite, or it's just injection, goes in the blood, becomes part of a serum, and then it works right away.
Oh, okay. So what if the dog, like doesn't have any rabies thing? So what happens to the antibodies then?
Then the antibodies will do nothing. It will just be in your body for a little bit, and then they get broken down. Right?
Okay.
Yeah. Just like the RhoGAM, right? The RhoGAM that the mother receives, right? If the baby cell does not go into the mother's body, then the antibody does nothing, right? Antibodies in general will not harm you, but there are some cases where it does, like in autoimmune diseases, which we'll talk about probably not today. I think I've expanded all your attention already. We are making good progress, guys. We will definitely be able to finish on time for the first unit. Okay. All right, active now. Active, we have natural, and of course we also have artificial, aka induced. So natural is very easy to remember. Okay, natural is, a person gets sick, again, like chicken pox, recovers, And now they have lifetime protection. Okay, e.g. chickenpox. Okay, so people nowadays, they can get vaccinated against chickenpox, but for me, I caught it naturally. I mean, I don't remember anything. It's just my mom telling me, like, when I was a baby, I caught the chickenpox, and then now I have protection. Okay, my aunt, when her son, I think this, yeah, when her son got the chickenpox, she deliberately infected the other sibling as well, right? Just to get rid of it, right? Just make sure that they also have the immunity, right? There are these things called like chickenpox party. I don't know if you ever heard of that before, but the point is try to, let everybody have the immunity. I don't know if that's like scientifically sound, but apparently that was a thing. So we as in like you and I, we have countless examples of natural active immunity throughout our lives. Every time you got sick and you recovered, you have immunity for whatever got you sick. Okay? Now, long story short, I'll explain this further next class. The flu virus, the COVID virus, those viruses, they are RNA viruses, as opposed to something like chickenpox, that's a DNA virus. RNA viruses, they don't have a spell checking mechanism, so they make mistakes every time they copy their DNA. And these mistakes allows them to evolve very quickly, whereas chickenpox virus, they don't evolve. So the chickenpox, we can get lifetime protection because it's the same virus that's circulating. They don't change over time, okay? That's why you get infected by the same chickenpox virus, no problem. Your body knows how to deal with it. But if you get infected by a different COVID virus, different COVID variant, right? You hear that on the news all the time, right? The Omicron variant, the whatever variant, right? The Delta variant. All these variants are actually different strains of COVID, if you would, and your immune system, the antibodies that you made previously might not work as well. Okay, so that's why you can catch some of these things repeatedly and not for others. So how do you protect yourself from getting too sick, too ill? Well, you can get artificial active immunity, right? Which is like a vaccination. Okay, vaccination. So e.g. a flu shot. The flu shot contains a weakened form of the flu virus. It activates your immune system. You go through the process of the T cell clonal expansion, choosing the B cells, and you end up having memory T cells and B cells as well as the antibodies. So when the real thing comes, you are protected. Okay, but sometimes the flu shots don't work very well. Okay, just a quick note here for you: the flu virus, there are many different strains out there. Okay, so on the surface of the flu virus, there is a H antigen and an N antigen. So like when we say H5N1 or H5N2, that's another one. All these things. There's also H1N1, right? You guys remember that was the previous pandemic in what was it? Was it 2009? I think it was 2009, something like that. Okay. That was a previous pandemic with the swine flu. It was called the swine flu back then, right? So all these H, all these N, that's talking about like the versions of these antigens. Okay. And so each season, flu season, the circulating strain of the virus is going to be different. Okay. And we have to make the flu vaccines ahead of the flu season, right? For us to be able to give it out to people, to the public. And so they as in like, World Health Organization, I suppose, or the CDC, Centers for Disease Control, they have to make a prediction of what the top 3 strains of the flu virus will be in the coming flu season. And then they make their best guess and make the vaccines for those 3 things. Sometimes they get it right, sometimes they get it wrong. It's just a prediction, right? And in years where they get it wrong, then the flu shot is not as effective. Okay. And remember, people keep on saying, like, my mom says this all the time, like, oh, I got the COVID vaccine and I still get COVID, right? Or like, oh, I got the flu shot and I still got flu. You have to understand the vaccine does not prevent you from catching the disease, but it makes you less ill, right? That's the whole point. You might still get sick from it for like, you know, a day or two, because it still takes time for your memory cells to kind of reboot, right? To mass produce those antibodies again. But the point is, you're not going to be as ill, and you're going to be able to recover quicker. That's the point of the vaccine. Okay, so that's why people need a flu vaccine every season, because it's a different strain every year. Whereas something like the MMR, right, or the chickenpox, they don't change. So if you had your MMR like 10, 20 years ago, you were probably still protected. So that's it for now. I mean, we still have a few slides to go, but I think that can wait. Okay, I don't want to, you know, over exhaust you guys. Any last minute questions before we go about anything that we learned today? No? Actually, you know what? We have a little bit of time to go through the lab quiz. Let me just show you what that is about. So when you go on eCentennial, you already have access to your lab quiz. Okay, so I'm gonna go here, quizzes, and the first lab quiz is gonna be due on June 5th. Okay, the day before the first test. And you will have 2 attempts for these ones. Okay, you can do it 2 times, and only the best attempt will be recorded. Actually, you know what? Before I show you about the lab quiz, there is the lab that you have to do. So if you go to content and you go to the lab, there is your lab 1. Okay, so you're gonna click into that. You do not have to hand this in. Okay, but you should download it and then you should do it yourself. So here it says, right, like if we were doing this in person, I would show you these models and then you'll be handling it in small groups and, you know, looking at it and answering questions. But this is online virtual, right? So label A, B, C, D. So does anybody know what A is? Maybe still remember from lecture 1? What is A?
Is that a aorta?
Yeah, that's the aorta, right? So you're gonna go through all these things, right? C is a coronary artery, for example. You write it down and keep it for yourself. Do not hand it in. So you're gonna do it for the heart, right? There's some blood typing stuff that we did, right? The respiratory system, you have to wait. I haven't teach you yet. Okay? But there's just, you know, some slides. Okay, 26 slides. You do it on your own, okay? And then you keep the answer. And then when it's time to do the lab quiz, you can use those answers to help you. So there will be some word question like these, okay? But then there might be like picture questions as well. Where is it? I don't know. I'm just randomly clicking through it. Yeah, so there might be a question like this. Okay, like what is the structure of H? Then you have to find the right answer, right? Okay, and this would be the same model that you would have seen in the oh, it's over here in the package right here. Okay, it's the same model, right? Right, it's just this guy, right? So that's why you have to go through the slides yourself.
The quiz just for lecture 1?
No. The lab quiz is 1, 2, 3, 4.
Oh,
okay.
wow.
Yeah. And so there will be questions that are based on these slide decks, these models, and then there will also be like, you know, standard multiple choice questions. It's not difficult. People usually get like over 95 on it, okay, because you get two attempts. It's more, sure, it's worth 2.5%, but it's more like a review tool for you for the test. Okay, because it forces you to go through these questions, right? And you know, help you review. Now, mind you, the test it's only 2, 3, 4. Right? Lecture 2, 3, and 4. But the lab quiz is everything, including lecture 1. Okay, so you can do that when you're ready. Like I said, 2 attempts, best attempts count. Don't do it now. Like wait until I finish teaching you the respiratory system, and then you just do it. And that's going to be the same thing for lab 2 and lab 3 and so on and so forth. Okay, the dates are all wrong for all the other quizzes and tests, right? Like just don't mind them. Don't look at them for now, because you know Miran has to set it up when he takes over the course. Okay, like I'm only here until test 1, right? So I'll set that up for you at a later time. Yeah, that's it. Any other questions? Great. All right, you guys did quite well on the first quiz. So keep it up, and I'll see you next Tuesday. All right. Have a good one.
Thank
Bye.
you.